In this video, we're showing you how we made our own OnePot pure solution for our 2019 EPFL iGEM project entitled ViTest. It consists of the buffer preparation, the energy solution, the protein solution, and the ribosome solution. However, the ribosome preparation is not shown in this video. Now we're adding 1 microliter of tRNA to 999 microliters of water. Be careful when using tRNA because it can be degraded very quickly by the RNases in the environment, so do not speak while pipetting. Then you vortex and centrifuge the mix. Next, you want to measure the concentration of the tRNA using the nanodrop. Don't forget to use water as a blank. Now defreeze the elements of the energy solution and vortex them quickly. Now you're going to add every part of the energy solution into one tube. Always keep your energy solution on ice. And always mix the solution well. And now you're done. Now we're going to aliquot our energy solution into 20 different tubes of 25 microliters each, because the reactions we perform with the OnePot solution are very small in volume. Don't forget to always put your energy solution in the -80 freezer. Next thing you'll want to do is prepare all the buffers as is stated in the paper. And make sure you measure the amounts properly. Use the appropriate tools for different weights. Make sure you dilute the powdered form of your buffers with bidistilled water. You can dissolve it by either using your hand or the magnetic stirrer. Do this for every buffer. Remember to pipe it very slowly for glycerol since it's very viscous. We use a hydrophilic filter from Merck because it has a 0.22 micrometer precision. We like to use this type of filter because it's more environmentally friendly for the lab. Now just pour your buffer inside. And use a low pressure environment to filter the buffer. Make sure to change the filter paper every time. For the glycerol, we're going to use a syringe filter. That's it, now you have all your buffers ready and labeled. First, make sure you autoclave everything. Then make sure you use the appropriate workbench and use a flame to sterilize the area. Here we're making the LB medium we're going to use and adding ampicillin. Here we inoculate the EFTU in 3 milliliters of LB medium. Next we put 0.3 milliliters of LB medium into 35 wells. It should look like this at the end. First, we will remove the aluminum foil from our bacterial stock. Using the replicator, we're going to inoculate the bacterial stock that was stored with glycerol into LB medium. Then we add a breathe easy membrane to our deep well plate to allow for aerobic growth. Then we use an aluminum foil to store the bacteria we used. Don't forget to label the wells properly. We then incubate our EFTU culture and deep well plate at 37 degrees overnight at 260 RPM. Now we're preparing 500 milliliters of LB medium with ampicillin for the large cell culture. We use the 1 liter buffled flask for the large cell culture. We take 55 microliters of each of the 35 protein cultures and put them in the 1 liter flask. Every time you pipette the bacteria, make sure you pipe it up and down so you get a uniform distribution. Then we add 1,675 microliters of EFTU into the 1 liter flask. Then we incubate for 2 hours. After the 2 hours are finished, we induct IPTG into the 1 liter flask. Then we incubate for another 3 hours. Then we harvest the cells by centrifugation at 4,000 RPM for 10 minutes at 4 degrees. Then you discard the liquid and store it at -80 degrees overnight. Remember to add again beta-mercaptethanol each time you reuse a buffer. Because you're using beta-mercaptethanol, you must use the chemical hood and wear glasses. You take 5 milliliters of buffer A and resuspend the cells in 1 tube. Then take the solution and resuspend the cells in the second tube. Then with 2.5 milliliters of buffer A, you wash the first tube to gather all the cells that are left and move it to the second tube. In the end, you have your resuspended cells in 7.5 milliliters of buffer A. It's very important to sonicate on ice, because you want to avoid the sonication to overheat your solution. We sonicate with 4 cycles of 20 seconds on, 20 seconds off pulse at 70% amplitude. Afterwards, we remove the debris by centrifugation at 15,000 RPM for 20 minutes at 4 degrees. We then added 2 milliliters of nickel NTA resin to the bottom of the column. The column was then washed with 50 milliliters of demineralized water and equilibrated with 35 milliliters of buffer A. Never let the resin dry out. After centrifugation, we put the supernatant into the column and we incubate it for 3 hours at 4 degrees. After all the unbound lysate has flown through, we wash with 25 milliliters of wash buffer. Finally, we elute the proteins using 5 milliliters of elution buffer. To do the buffer exchange, first we mix the 5 ml of eluted solution with 25 ml of HT buffer. Then we load half the solution, 15 ml, to an Amicon filter with a 3 kilodalton cutoff. We then centrifuge it for 60 minutes at 4000 RPM at 4 degrees. Do the same thing for the other half of the solution. You should expect the final quantity to be 1 milliliter. Finally, we dilute with 10 milliliters of HT buffer and centrifuge for 1 more hour until we reach to a final volume of less than 1.5 milliliters. Here we transfer the solution that didn't flow through into a tube and mix it with an equal amount of stock buffer B. The final step is to concentrate the protein solution using a 0.5 milliliter Amicon filter with a 3 kilodalton cutoff, which we centrifuge at 14,000 RPM for half an hour at 4 degrees. Our OnePot protein solution can then be stored at -80 degrees.